All right, I'm going to turn up the microphone. Can you guys hear me okay? Sorry, my voice is all jacked up. Some stupid virus. But hate that. All right, so today we're going to talk about replication. Remember we were doing DNA replication, whatever day that was, Thursday? DNA replication physiologically is for when cells need to reproduce themselves, right? If you need to make 2 more cells because you got a cut and your skin needs to heal over, or you need more liver cells or brain cells, which would be nice for us, or if your immune system needs to sort of, make some more white blood cells to fight whatever virus happens to be taking over your mucous membranes. Then you need to first do DNA replication so that the 2 cells that come from the original have the same amount of DNA, right? If a cell starts with 46 chromosomes and then it splits in half to make 2 cells, you want each of those cells to have 46 chromosomes, right? Every single cell in our body needs to have all the genes, all 46 chromosomes, 2 copies of every gene in all of our cells. The only exception to that is in eggs and sperm, okay? Those are the only 2 cell types that have half the amount of DNA. Everything else needs all 46. So what evil scientists have done is taken the process of replication and repeated it in a test tube, okay? The polymerase chain reaction is just DNA replication, but done in a test tube instead of in your cells, okay? So we figured out all the enzymes that are needed to do DNA replication, all the other stuff, right, all the nucleotides, all of that crap, and we can do that now in a test tube to amplify or make more DNA. Okay, so the process of PCR, which is the polymerase chain reaction, which we're going to talk about, has revolutionized our ability first to just study genes and so forth, in science for health and all that, but also for personal identification of individuals. Okay? So we're going to talk about PCR and then how it's used to solve crimes and to figure out who's the daddy and all that kind of stuff too. Okay, so we talked about in the video of replication from last week Thursday, the real one, the educational one, I said you guys don't need to know all the details of replication, right? You need to know that it occurs by complementary base pairing, right, on both strands of DNA, right? Every single chromosome is really double-stranded DNA, a big, huge, long piece that then's all wrapped up on itself to make that little sausage guy, right? The 2 strands stick together because of complementary base pairing, which is A's and T's stick together, G's and C's stick together, T's and A's, C's and G's, right? All the way across. We separate that, and then that enzyme DNA polymerase adds nucleotides by complementary base pairing. If it sees an A, it adds a T. If it sees a C, it adds a G. If it sees a T, it adds an A. If it sees a C, it adds a G, and so forth, all the way down to make an exact copy. Now, in that little video, they talked about you need a primer to do that. The little piece of RNA has to stick in there to get it going, and all this other baloney that had to happen, which you guys don't need to know for regular DNA replication. Okay? But in order for scientists to come up with a way of doing this in the test tube, they did have to understand exactly how it works. And what does that DNA polymerase enzyme need, right, to actually do replication? And so in order to do replication in a test tube, right, you need to have those free nucleotides, right, the A's, the G's, the C's, and the T's, those molecules, in order to add to do complementary base pairing, right? Those have to be available, just like in regular replication, right? We need this same DNA polymerase enzyme, right, because that's the thing that does that actually adds the base pairs to build that DNA molecule. And then we have to have primers to get it started. And those are, in this case, are just little pieces of already made DNA that's complementary to your template. Okay? So for PCR, we don't have to make the entire chromosome. Whereas in DNA replication in a cell, right, they're replicating the entire chromosome of all of our chromosomes. If we use PCR, we can replicate just a small part of a chromosome just by where we place these little primers, right? We know the entire sequence of the human genome, right? We talked about that last time. We've sequenced the whole genome. We know what every single base pair in order on all 46 chromosomes. So any genes that have already been identified, like insulin, for example, we could just design these little primers to be complementary to either side of the insulin gene, one going that way, one going that way, and we can do DNA replication of just the insulin gene, okay, using this PCR. So we can make a whole bunch of that insulin gene DNA, and then we can study it or use it to do other stuff. Okay? So for PCR, we don't have to make the entire chromosome. For regular DNA replication that happens in your cells, we do want to make the entire chromosome. But here we can just make individual genes or just small parts of the chromosomes, just by how we make these complementary to that little sequence. So we can sort of target what genes we want to do with PCR. Okay? But we still need all the stuff excuse me, that the cell would need to do replication in order to do this process. Okay. Okay. And so here's our little test tube that we do all this in. First, we need DNA from we need the template DNA, right? So we need something that we're going to do this PCR off of, right? So if I wanted to PCR out one of your genes, right, I would need to take some cell samples from your cheek, right? If you just swab the inside of somebody's cheek with a Q-tip, you can get enough cells on there to extract the DNA, and then we could stick it in a test tube to do PCR. Okay? And then we add our nucleotides. Remember, these are the bases, the nucleotides A, T, C's, and G's, those molecules, those are the ones that we stick in there to build up. DNA polymerase enzyme, right, which has been purified. In this case, we use enzyme from bacteria because it's easier to get from them. And if we go back to all the crap we learned about evolution, we all use the same DNA as our genetic material, bacteria and us, right? The exact same nucleotides, almost the exact same enzyme. For our purposes, DNA polymerase in a bacteria works exactly like the DNA polymerase in your cells. So we can get it from a bacteria, right? And we can get it from a bacteria that lives someplace like in the ocean in those thermal vents where the water's boiling. And so that enzyme is very stable even at high temperatures. Most enzymes totally suck at high temperatures or low temperatures. They die, they fall apart, they don't work. So ours, if we extracted it from your cells and had a bunch of this enzyme and tried to manipulate it in the lab and students carrying it around and it's not on ice or it's not at 98.6, it wouldn't work anymore. But we can get it from a bacteria because we all use this same system to do replication. So we use a bacterial excuse me, polymerase, and then we have to add these little primers, little pieces of DNA, AGCT, about 20 base pairs, little tiny piece that's complementary to the region wherever we're trying to amplify. Okay? Instead of doing it from the ends of chromosomes and amplifying a billion base pairs, we can just do PCR on 300 or 1,000, just a small piece, right? Just the insulin gene or whatever we're looking at. And so then here's our little picture. Again, I want you to understand conceptually how PCR works without me necessarily asking you specifics. You should know that it needs the DNA polymerase, it needs the primers, it needs the nucleotides, and you have to have a template. The same exact stuff that we need for regular replication. Okay? So we can make billions of copies of this little piece of DNA from a very small amount of starting material, right? If we scrape just a little bit of cells from your cheeks, we can get a tiny bit of DNA. You could never see it, right? We'd do a DNA extraction on those cells, and at the end it would look like, you know, a couple drops of water. And then, you know, you okay, well, hopefully it's in there. And then from that, you know, teeny tiny little few drops of water, you can make enough DNA so that you could actually see it look like a white little stringy thing in a test tube. Okay? If you take enough cells, like if I took an entire strawberry and mushed it up and extracted DNA, we could see it. It looks like spider webs because there's so much in there. But that's because a strawberry is made out of 1 billion cells, right? If you're only taking 30 cells from your cheek, you're never going to see it. So a teeny tiny amount that we can amplify, because all we really need is one molecule of DNA to get our primers to sit on. DNA polymerase will make a copy of that, and then we do that cycle again. We do replication again, and then we make copies of that, and over and over and over and over. Okay? And so then every time you make a new little piece, the primers can sit on that and amplify the original template and the newly made one. And so if you run it through 30 cycles, you can make billions of molecules of DNA from a very, very little bit that you'd start with. Excuse me. And so to do this, they just heat it up, they cool it down, they heat it up, they put it at the different temperatures that the enzyme works, right? We get the primers to sit, the enzyme sits on there, it does complementary base pairing, we heat it up, everything falls apart, right? Just like in replication when the two strands come apart, right? Then we can have the primer sit, do DNA polymerase, complementary base pairing, heat it back up, fall apart, and over and over and over and over, so you can make tons and tons of a little piece of DNA, okay, from 1 or 2 molecules of your sample. And so what we can use this for, one of the main things that PCR is used for now is for crime scene identification of tissues or fluids left behind, right? 1 drop of blood you can get enough DNA from to do PCR to identify the individual that that blood came from. Okay. 1 strand of hair, you can get enough DNA from the little piece of skin that comes out of your scalp to do PCR and identify the individual from where that hair came from. Okay. Now, if you just cut somebody's hair, your hair, the actual hair part is just protein. Okay, there's no DNA in this, right? It's like kind of like your fingernails. There's no DNA in that. But if you, okay, everybody take 1 strand of hair, and now yank it really hard. Nobody's doing that. Come on, people, work with me. Okay, you'll see that little white piece at the very end. They call it a skin tag. That's just a little piece of skin that yanks out when you pull it from the roots, right? You pull a piece of hair out by the root. That little piece is some skin cells. That's enough for us to isolate the DNA, do PCR, and determine that hair came from you or from whoever else happened to be in the room. So if you watch CSI or NCIS or any of those things, they're always collecting hair samples, right? But it has to have that little skin tag. So you can't just cut, you know, a piece of somebody's hair. But if you look at somebody's hairbrush, right, if you're brushing your hair, right, your hair might be a little tangly, and so some of it gets yanked out, right? And so there will be some on your hairbrush that has that little skin tag. So they'd have to take those pieces, not the ones that just break or, you know, have fallen out and they don't have the skin on it. So it has to have that little skin left to get DNA from. They could collect saliva, spittle, right? Because the inside of your mouth, you're constantly sloughing cells off of your cheeks. That's why if you just rub it a little bit with a Q-tip, you can get a bunch of cells on the end of that. So somebody's saliva has enough cells in there, 3, right? You'd never see it, but that's enough to get DNA to do PCR to then identify who that person was. Okay? So it's like fingerprints, but on steroids, right? It's your DNA fingerprint. You guys are all unique on this planet. Nobody else has exactly the same DNA as you unless you have an identical twin. Right? If you have an identical twin, you will not be able to tell the difference between yourself and that twin using any of these DNA tests. So if you have an identical twin, make sure they're not evil. Okay? If they're good, then you're fine. But if you have an evil identical twin, then that's a problem. Okay. So DNA profiling is just taking advantage of this fact. Being able to collect a small sample from that individual person, do PCR, sequence it to see exactly what base pairs are in that area, right? And then identify the person that it came from by taking a sample from that person, right? So on all the, law and orders are always, you need to give a DNA sample, I need to swab your cheek. That's so they have a reference to whatever they found at the crime scene. And then they try to match it by doing PCR and sequencing or fingerprinting. DNA fingerprinting. Okay. So how does that work? When I was talking about evolution and I said everybody's genes are very similar to each other, right? To the chimpanzee, most of our genes are like 98% identical, right? I was just Googling and being nerdy, and I was looking up the insulin gene because that's something everybody can sort of understand. And the chimpanzee insulin gene is like 99.4% identical to ours. Okay, that's kind of crazy. So then I decided, well, I would look up a mouse. What is mouse insulin? Mouse insulin is like 84% identical to humans. That's pretty crazy. So then I went like to some crazy platypus organism, and it was still like 72% identical to humans, right? So we're not all that different from the platypi. Although, a fun fact about the platypus that maybe you guys don't know, a platypus has a forked penis. What? Those people in the back just perked up. Yeah, it's a forked penis, people. Which means the female platypus has 2 vajayjays. Dual action. Okay. Sorry. Fun facts. I don't even know what I'm talking about now. Okay, I got all distracted. Must be the cold medicine. Okay, so the coding sequences, or the genes that actually make stuff like insulin, neurotransmitters, DNA polymerase, those are really similar from person to person. So if I just did PCR on insulin gene from you, I can't tell you guys apart, okay? You guys would look like evil identical twins, right? But there are regions of our chromosomes that are very different from each other, okay? And those are the places that we would do PCR, and then it's very easy to tell the difference between two individuals. Now, if I took your entire genome, all 46 chromosomes, and sequenced them, and all 46 chromosomes and sequenced them, yeah, it's easy to tell you guys apart. But that's, $20,000 a piece. Our, LA County police stations can't spend $20,000 to do a genome sequencing to look for the evil twin or the crime scene guy, right? But PCR of a region that's very different between individuals and a bunch of different ones is really cheap, right? That's like $10 in supplies, and then however much an hour they're paying the poor student that's doing the work, right? So that's cheap. That's totally doable on a county budget, right, or a city budget, right? So we look for regions, non-coding, things that don't make proteins on our chromosomes, right? The structural regions of like the middle, where they stick together, that little pinchy part in the big X of the chromosomes. That stuff is really different from person to person. And so we can make a genetic fingerprint from that, okay? So then everybody's, as long as you look at enough different chromosomes, right, everybody has their own pattern. Kind of like fingerprints. Everybody has their own pattern of fingerprints, right? Again, unless you're an evil twin. We can get a genetic fingerprint, a DNA fingerprint from individuals, which now is pretty cheap. It's pretty easy to do. It's totally doable for an everyday sort of crime scene investigation. Okay. So these are just called STRs, and they use the differences in lengths of these between individuals to distinguish between them. And if you look at 10 different chromosomes, right, they do the statistics where the odds of 2 individuals having the same pattern, if they look at 10 or 12 or however many they look at, is essentially 1 in 10 billion. And if there's only 7 billion people on the planet, right, you're not going to duplicate that. So they just crunch the numbers in some crazy math algorithm to figure out how many regions they need to look at to make sure they're very comfortable that it wouldn't be a mistake and that they would get the wrong person using DNA. So this is just an example. So this individual in that structural region has repeats, 6 on one chromosome, 4 on the matching chromosome 1 and chromosome 1, right? We have 2 of each, right? This guy looks different than that individual. And so everybody would have sort of their own pattern. And then if we looked at 10 or 12 different chromosomes, right, for each person, it's extremely unlikely, 1 in 10 billion chance that 2 people would have that same exact pattern. Okay. So you can identify them without spending $20,000 to sequence their entire genome, right? So you could spend $10 to $20 per person to make this happen. I could do this in my lab upstairs with undergraduate students, even you guys, right? Because I know you love biology. You want to come to my lab and do PCR. Yay! Okay, so this is a technique that we do all the time. Not for solving crimes, but for other reasons, okay? So again, unless you're identical twins, you have unique DNA. The process of DNA fingerprinting or DNA profiling, I think DNA profiling is what our book calls it, right? So first you have to get some of the DNA, right, from whatever the unknown is, and then compare it to the possible suspects if you're doing a crime scene, right? So anything that might have cells in it, you can extract DNA from. And you only need a teeny tiny little bit because of the power of this stupid complementary base pairing doing PCR, right? So the template, what you're working from, very, very little amounts of DNA, right? So when you see them taking saliva from a glass, at a crime scene, or one hair with the little root, skin tag, that's not bullshit. Lots of stuff on TV is total bullshit, right? But that's not. Okay, one little tiny drop of blood. Even dried blood will work. DNA is really stable, so you can get DNA out of dried blood. Okay, you might not be able to get like all 46 chromosomes intact to sequence and get an exact, every single base pair of their genome, but you'd have enough to do PCR to make a decent profile. So even a tiny little piece of dried blood would work. Right, skeletal remains. And like we talked about last time, we can't use fossils, right, because fossils are really just rocks. There's no cells, there's no DNA left in them, unless it was mummified. Mummified tissue, sometimes you can get enough DNA. Mummified is like freeze-dried almost, right, and so sometimes there's enough DNA in there to get an extraction to work, right, or frozen. Right, the caveman frozen in ice type thing, you can definitely get DNA from that and do this kind of profiling. Okay, so here's our little, first we have to collect the cells and extract the DNA, right? The easiest thing that they do if they're getting cells from suspects or to eliminate people from being a suspect would be just cheek cells, right? You just do that little swab. It's super easy. They come right off. you just use a little kit to extract the DNA from that, get rid of all the other stuff, and all the membrane proteins and all the organelles and all that crap that I tortured you guys with a couple weeks ago, right? So all we have left are those chromosomes, is the DNA in the, busted open the nucleus, all that stuff. Okay. Then we do PCR on a whole bunch of regions. They're giving us 3. It's more like 10 or 20, but we'll just, do this little example. Start with a teeny tiny little amount, right, and then amplify a whole crap load, right, so that you can actually do something with this and be able to visualize it with the naked eye, right, so that you don't have to do anything crazy and spend a ton of money to determine what's in here. So this is just another picture of what PCR is, and it goes through cycles. So if you have your template, right, and then you separate it, we do DNA replication on both sides, complementary base pairing, A's and T's, G's and C's, right? And then we heat it up, separate them again, do complementary base pairing on each of these strands to get these, and then heat it up again, do complementary base pairing on each of these strands. Right? And so this would be exponential amplification of DNA because you double each time. Now all 4 of these are going to open up, and then all 8 of those, and all 16. And so over a course of 30 or 35 cycles, you get a ton of DNA. Right? You'd get a lot if you started from 1 molecule, right? But if you have a drop of blood, you might end up with 100 molecules to start, and you're going through this exponential amplification, making a whole lot more. Okay? So you can get lots and lots of DNA just from what we would think is a little tiny bit. It's really just replication in the test tube. Yes, sir. Sorry. His question is, would you be able to extract DNA from a burned corpse? As long as it's not completely down to ash. So normally if somebody burned in a fire, there would be at least some bone left, and then you can definitely extract DNA from bone. So if they're cremated down to there's just ash, no chunks left at all, then no, we wouldn't be able to get it from that. But any sort of burned victim, in a burned out car or something like that, it would be rare for it to be to a point where there was no tissue available. Yeah?
But don't they have like 2 different fingerprints?
So identical twins can have different fingerprints. So fingerprints come up during development, and they are based on environment and DNA. And so again, identical twins, if you sequence their entire genome, they may have a few base pair differences, mutations that happen, throughout their lifetime. But doing something like this, where we're only looking at 12 places, we wouldn't be able to tell them apart. But sometimes, yeah, you can tell by fingerprints. It just depends on, because fingerprints they are coded for, the development of the skin is coded for by DNA, but there is some environmental influence on what your fingerprints actually look like. That's a great question. Thank you. Okay. So are we good with PCR? Amplify DNA from a small amount. Make a whole lot more. Okay? Okay, so what do we do with all this DNA once it's in a test tube? Again, we could sequence it and look at the base pairs themselves, AAT, GGC, CCC, that sort of thing. But again, that's expensive. So if we want to be able to look at DNA and do it relatively cheaply, some way that it's possible to do in a crime lab that doesn't have an enormous budget, we can actually look at DNA by how big it is, right? By actually how much did you amplify. And so the picture that we saw earlier where some people's region is very short, some are middle, and some are much longer, right? We can actually determine the size of all these little pieces that we made by PCR by using something that they call gel electrophoresis. And the gel stuff, the matrix that you run that DNA through, is essentially Jell-O, like the yummy Jell-O that we eat, except there's no sugar or artificial food coloring in it. Okay, but it's that same stuff. So if you've ever had Jell-O, right, it's all wiggly and it's kind of thick. We pour Jell-O into a little pan, right, kind of like the Jell-O jigglers, and then you can cut little holes in it, and you put the liquid that, there's all this DNA in there that we can't see, right? We can stick it in so that it's stuck inside this block of Jell-O, and then we hit it with a whole bunch of electricity, right? So what happens if we use electricity, right? Even if we don't know anything about electricity, electricity is just positive and negative charges, okay? Ions passing through positives and negatives, that's all electricity is. So if we pulse it with a negative charge up here and a positive charge down here, the DNA molecule itself is negatively charged, and so opposites attract, so the negative charged DNA will travel towards the positive charged electricity that you're sticking this through. Okay? So it's in a watered aqueous solution. If you turn it on and you stick your fingers in that water, you get a big shock. Okay? So it's real electricity. So you have to be careful that that doesn't happen. Okay? And then because this is that thick Jell-O stuff, it's really hard for the DNA to sort of migrate through, to get through that thick, gooey matrix. And so the small pieces get through a lot easier because they're little and they can work their way through all that Jell-O. And the medium pieces are the next fastest through. And the big pieces get stuck up here. They can't travel through that Jell-O. Right? So you can think of it like if this whole bottom part of this room was a Jell-O jiggler, right? We filled it this deep, and then we have some little 5-year-old, well, maybe they have to be tall enough to have their head out, right? A little kid, a little scrappy kid, and then a big, giant football player guy, big fat guy, right? The little kid can sort of dart through this thick matrix much more quickly because they're small, right? Where the big guy's got so much surface area that it's really hard to get through this thick matrix, okay? So the big fat football player is the long pieces of DNA. They can't fight their way through. So they get stuck up here in the same amount of time. If we stick them all in one hole, we turn on electricity for an hour, the big ones stick up here, the medium ones go down here, the smallest ones run down here. So we can separate DNA by size. Okay? The small guys run towards the bottom, the bigger guys run up here, and then the pattern of all the different suspects, we PCR'd off of 10 different chromosomes, right? Their pattern of all those pieces will be separated by size. And every individual, unless they're an identical twin, will have a different pattern of what those look like. So if you have the saliva sample from the crime scene and 3 suspects, the person that left the saliva would have this exact same pattern. None of these guys have that pattern. All of these guys are innocent. None of these guys could have possibly left that saliva because their pattern of DNA is different from this. Has to be exactly the same for it to be from the same person. Yeah?
But, I mean, by the time the saliva collects saliva, doesn't it dry up? Or?
Yeah, you can collect dried saliva, and this, even if the cell's dead and dried up, the DNA can be intact in that.
How would they know how they find saliva is dried?
Well, so on the stupid show, the question was, how do they find saliva if it's dried? If you've seen, they offer the suspect that won't give DNA a glass of water, right? And then he drinks it, and he's apparently a big slob, right? And so then they collect that off the rim of the glass. They would do that at a crime scene. You see, you see a table set and there's glasses of wine and then there's a dead guy on the floor, right? So they collect the glasses and they put them in bags and then they try to extract the saliva off the cups. That would be one way, right? And then they just take a DNA sample from the dead guy. They just do a cheek swipe on the dead guy, right? And then they could figure out, well, which wine glass did the dead guy drink out of? And then the other one is, oh, somebody else was there. Who was that? Right? And then they collect the suspects and they cheek swab and they do PCR, right? And then they run it through this agarose and they look at this stupid banding pattern to see if any of the suspects match the wine glass DNA. And in this case, they don't, right? So they didn't catch the bad guy yet. Does that make sense? Yes, sir. Sorry. They would be able to use any dried saliva, but they would want to make sure that the glass, both of those glasses, one of them came from the dead guy, right? Because maybe the dead guy wasn't drinking any wine and there was two people there. So, they'd always take a DNA sample from the victim to make sure any other DNA, there's DNA all over the place to, if there was blood, that, okay, that came from the victim, that's not a suspect, right? To try to eliminate all the samples that just came from the victim, okay, if you're talking about that kind of identification. Okay. So this is just the summary of what I said about the gel electrophoresis, right? And so it's just based on the charge of a DNA molecule, which just by chemistry happens to be negative, right? You don't have to care about that, right? So it runs towards the positive charge that we put in there. So if you think about it like if you've ever had a car that you need to use jumper cables, you put the red on one and the white on the other. One is positive, one is negative. You start the other car and you jolt that battery to get its charge back. It's the same idea. There's red on one, white on the other, and then we turn on the electricity and it sends those charges that way. The positive we put down at the bottom, the negative DNA will travel in that direction, okay? And the small pieces go faster because they can just scoot right through that thick, jello-y crap. So the big guys are way up near the top, the small guys are at the bottom, so you can get this nice pattern. And everybody's pattern is different from each other. And then I just said all that too. Sorry, talking ahead of myself. So this is a summary of what we talked about. So again, when you get home and you are studying, and you go, oh my God, I can't remember what the hell she was talking about, that jello crap. Here, it's all written out for you, right? So you can always look in the lecture notes and look at that. And they just call it gel electrophoresis because it's gel, it's a jello thing, and we shoot through electricity to separate DNA molecules. Okay. And so then as we're doing that, we can't see that DNA, right? We still, in the little tubes, we still, even though we went through all those rounds of PCR, we can't really see DNA. And even if we did, it would just look shadowy like white stuff, right? We couldn't tell the sizes of it. We don't know anything about what it looks like. We run it through this gel stuff. We have to actually use some sort of fluorescent stain to see those banding patterns, right? Some kind of dye or some kind of stain that particularly grabs onto DNA and then fluoresces so we could take a picture of it. So then we can actually see bands for each lane, right? Ooh, as we're running it, it just looks blank. So up until the point where you stain it, you don't really know if you totally screwed something up somewhere, right? Because you isolate DNA, it looks like water. You do PCR, it looks like water. You load the gel and run it. It looks like water. And then at the last step, you stain it, and you either are happy because there's bands there, or you're like, oh, crap, I did something wrong somewhere, and I have to do it again. Okay? And so this just shows you, right, that in order for, excuse me, in order for one of the suspects to have really left this behind, every single band has to be exactly the same, right? The odds are that some people may have one or two bands similar to each other, about the same size, right? But by probability, the number of chromosomes they look at, nobody on the planet has the exact same pattern of DNA, okay? So just because these guys all have one down there, but they don't look, well, sorry, this guy does. These two don't look exactly like that one, right? These guys are not the problem. Sorry, they're showing a different gel than last time. In this case, this one has the bottom blue, the middle red, another blue, and two of the greens up top. And they're just showing you these in different colors to help you identify the bands. Normally, they're all stained the same color. And we just look at them by size, right? And so the banding pattern of the saliva sample and this suspect letter C are exactly the same. And so they can say suspect C left behind that saliva sample. That only proves that the guy was there or at that location leaving behind his saliva. Doesn't say anything about whether he actually did something to the victim, right? But puts that person at the crime scene, at least at some point. Right? They've been to that person's house or wherever this happened. So even if they wore gloves to try not to, and shave their heads so don't leave behind any fingerprints or hair or whatever, right? If they drank out of a cup somewhere, then they can get busted. Okay? So in this case, let's see, I have a bunch of other examples so that you can get used to looking at these kind of things. So for the quiz, pictures don't come through well, as it turns out. At least most people's won't load. But for the exam, I'm going to show you a picture of banding patterns, and I'm going to ask you something about it, right? Like, who done it? Or who's the daddy, right? So in this case, we can use excuse me, DNA fingerprinting or DNA profiling for crime scene, right? That's one of the main things that we use this for. We can also use it to identify body parts, okay? So one of the biggest projects where they used DNA profiling was in the attack of the Twin Towers in 9/11, right? There were hundreds, thousands of people missing and lots of body parts everywhere, right? And families like to recover the parts of their family members. So they wanted to be sure that the foot goes, these 2 feet go together with this whatever and so forth. And so they took tissue samples of all the parts that they found and they did DNA profiling to match individuals together, to return them to their family members, okay? And family members, siblings share banding patterns, and parents to offspring share banding patterns. Okay, I'll show you an example of that too. So if they get a DNA sample from a parent of a missing person, right, they could use that person's DNA profile to determine which parts belong to that family. Okay, and so in this case, you can see the exact identical banding pattern of 1, 5, and 18, right, from the same individual. And so those bone fragments all came from one individual. Same with any kind of mass grave that's uncovered. They can figure out what bones go to what individuals. Okay. Again, as long as you can extract DNA from a sample, you can do DNA fingerprinting or DNA profiling. Okay. So here's an example of what a parent to child would look like. And so in this case, this was from a grave site where there were 5 adult skeletons found and 3 child skeletons, and they were trying to figure out if any of them were family members. And so what they did DNA profiling on all of these, and they put together the arrows to show you which bands match. So remember, the children have half of the DNA from their mom and half of the DNA from their dad. Right? Half came in the egg that made you, half came in the sperm that made you, right? And so half of your banding should look like your mom's, and the other half should look like your dad's, right? So you can do paternity by doing DNA fingerprinting as well, right? And so they show that adult number 1 and adult number 3 were the parents of child 1, 2, and 3, based on the banding patterns, right? So if you look at child number 3, that's probably the easiest. The 2 lowest bands match adult number 3, right? They're the exact same size. And the 2 small guys towards the bottom. And the 2 top ones match adult number 1. Okay, so half of the kid's DNA came from adult 3, the other half came from adult 1. Right? Now, a real fingerprint might have 25 or 30 bands, but they're simplifying it so that we don't have to keep track of all of that, right? Because you really couldn't tell from this few bands. Like, they would always test more places on chromosomes. But just for simplicity, you can see that that they come from the same 2 adults. And then child number 2, right? This band is one of the middle bands, and then the top 2, right, are from adult number 3. And then this child, top 1 to 1, next one to 3, and then the middle one to 3 as well. And then if you just look at the siblings, they do, in some cases, share some of the same banding patterns, but in others, they don't, right? They usually share a few. So if we looked at 20 sites, yes, siblings would share some sites. Any of the same chromosome that they got from mom, right, would have the same size band. If they got the same chromosome from dad, the half they got, then that would show up there too. So you can determine parent and child relationships, and you can also determine sibling relationships, right? You can determine family relationships, cousins and so forth, but only when you're looking at lots and lots of sites, like 20 to 30 different sites, to look at similarities, to build some sort of big family tree. But it is possible. Does that make sense? So that'll be something I'd ask you to do. I might ask you, show you a bunch of adults and say, who's mom and dad or child number 1, or give you one. Adult number 1 is this kid's mom, is, 2, 3, 4, 5, the dad. Okay? So applying what you've learned, right? Not just telling me what PCR is and what DNA profiling is, but being able to analyze something showing that you really understand how it works and how to look at that. You guys are all sleeping. Okay. Oh, we're almost ready to have some fun. Okay, so when we're talking about I'm sleeping too. Evidence for in criminal records, DNA is the most efficient and reliable method, as long as the samples were taken appropriately, like the police didn't mess it up and mislabel something, or contaminate their own DNA on top of whatever the sample that they're getting, or the crime scene investigators, whatever, right? You can identify people by their bite marks, but it's not, or their dental records, but that's still not as accurate as using DNA. So DNA is the gold standard for identifying individuals, right? Again, except for identical twins. Okay. Are you guys ready for something? Is it educational or not? I hope not. Okay, well, it is, but it'll be fun. Are you ready? I don't think you're ready.
There was a time when to amplify DNA,
You had to grow tons and tons of tiny cells.
Oh, and along came a guy named Dr. Carey Morley.
Said you can amplify in vitro just as well.
Just mix your tablet with a buffer and some primer. Nucleotides and polymerases too.
denaturing, annealing, and extending.
Well, it's amazing what heating and cooling and heating will do.
PCR, when you need to detect mutation. Detect mutation. PCR, when you need to recombine. Recombine. PCR, when you need to find out who the daddy is. PCR, when you need to solve a crime. Solve a crime. PCR, when you need to detect mutation. Detect mutation. PCR, when you need to recombine. Recombine. PCR, when you need to find out who the daddy is. PCR, when you need to solve a crime. Solve a crime. PCR.
To all the scientists out there doing PCR, Biorad salutes you with the all-new 1000 series thermal cycling platform.
Oh my goodness. That makes me happy. Oh my goodness. Look what time it is. What do we have here?
Oh, you guys made me eat.
Oh, so cute. All right, top hat people. Get out your phones. Oh my God, I don't even know what. Oh, it's supposed to be PCR. What do you think that is? Fascinating. No. No. Sex? The hell? Gonads! Oh, you guys are killing me. Huh. What about this? What the hell? Where's my PCR question? Is it in this one? No, we already did that one. It can't be cell cycle. No. I'm checking all of them. Oh, there's another. Huh. Fascinating. And what about down here? No. Okay, talk amongst yourselves. Here's the topic. It's lost. Mendel? No. No. I'm gonna cry. All right, I'll have to save it for next time. I don't know what the hell I did with it. It's not top hat time. I lied. I'm such a fat liar. Okay. What time is it? Oh, it's not time to go yet. Darn. All right, we will do that next time. Sorry. Oh, I know I'm killing you, aren't I? What's happening? What do we got going on here? Oh, okay, that's not too bad. That's not bad. It'll be okay. Uh-oh. Maybe we won't. Oh, there we go. Hello? Maybe we won't be continuing. The hell? Oh. Oh, I know, you're disappointed. Oh. She's gonna keep talking. Yay. Okay. What the hell? All right, we're going to talk about genetic engineering, which is really just what we've been talking about already. It's genetic engineering is anytime we really manipulate DNA to do something that we need for science, right? So a genetic engineer is anybody who essentially does PCR. Okay, so PCR is sort of the cornerstone of all the techniques that a genetic engineer needs to use. Okay, so anytime we isolate DNA by PCR, we call it recombinant DNA, because it's not just taking the chromosomes out of a cell. We just call that DNA or genomic DNA, right? We've actually done replication in a test tube, and we call it recombinant DNA. Okay, so a good example of what a genetic engineer can do, or why PCR is so powerful, and why this technique has revolutionized medicine and science. And we're going to use an example of insulin for diabetes, because I think everybody has a good idea that if you're diabetic, you're not making the protein insulin. Insulin's job is to allow sugar to get inside your cells so that you can use it for energy. So if you don't have any insulin available, the sugar doesn't get inside your cells, you can't make enough energy, you get very tired, and you can faint. Okay? And so if you're diabetic and you're not making insulin, you have to take insulin either orally or by shots, right, to supply your body with that. If you don't do that, essentially you die. Okay? So you absolutely need that to help you. And that's for type 1 diabetes, right? The genetic type, not the kind you might get as you get older because you're obese. That's a whole different story with insulin. Okay. So in that case, if you have type 2 diabetes, usually people are making enough insulin. Your cells are just no longer responding to it because for whatever reason, and they suspect it's due to high sugar load, it's like overloaded the system and it can no longer respond, right? And so you have diabetic crises, just like a diabetic that's not making insulin. And so for type 2 diabetes, you generally need to lose weight and eat less sugar, and all the symptoms will go away, your cells will respond to insulin again, and you're cured, okay? If you have type 1, where the cells in your pancreas are just not making insulin, there's either a mutation, those cells have been destroyed, you know, you're born with some genetic change that your insulin just got screwed up, right? You can help manage it by eating less sugar so you don't have so much fluctuation, but that doesn't cure you. You have to take exogenous insulin. And what used to happen is that insulin would either come from people, right, donate blood, and they would isolate the insulin from that, which you get a very little bit out, so it would be extremely expensive. And so a better source was to get insulin from a horse, right? Horses are big, strapping animals. And again, because we all are from a common ancestor and horses are mammals, they're more similar to us, right? That insulin from a horse can function pretty well in people. Not as good as human insulin, but pretty darn good, right? So there were farms where horses were just donating blood, right, to isolate insulin. Now, this didn't really hurt the horses. As it turns out, you can get a shitload of blood out of a horse, and he's totally fine, right? And then you don't take blood from him for another 2 or 3 weeks, and he's all happy on his little farm. And then he goes in and gives blood again, right? It's not painful. But, you know, there's just this whole farm full of horses, and that's what they do. They just give blood to have insulin isolated from it, Okay? So that's pretty expensive, right? And it's not as good as human insulin. So after the advent of this PCR and the ability to then take that DNA, we've isolated the gene for insulin from humans, we can take that DNA and we can actually make a bacteria make that protein for us, right? We can stick it inside of a bacterial chromosome, stick it inside a bacteria. We can grow up a gajillion bacteria in a big tub, right? Because they're too teeny, tiny, tiny microscopic, right? And they're just making insulin. And then you isolate the insulin from that, filter away all the other bacterial poop and cells, and now you have pure human insulin, right? You can make pretty cheap, and it's real functional human insulin, almost identical to the stuff you'd get from human blood, right? But super cheap and lots of it. So no more horses have to wander around spending their life giving blood. Okay, they just get to be happy and do whatever horses do. Okay. And again, because bacteria, right, use DNA and use all the same processes that we do for making proteins, right? The whole recent common ancestor from this planet, it makes insulin almost exactly like our cells would, right? Except way more, because you can have a cajillion bacterial cells, right? And much cheaper, right? Bacteria really need a little salt and a little sugar, and they can grow and make insulin for us, right? We could culture human cells and have them make insulin for us as well, but it's more expensive, right? It's harder to do. It takes more equipment, takes more media, takes more space, and the bacterial insulin is just as good. And so that's what we do. So that's the crux of genetic engineering, taking a human gene, sticking it into a bacteria, and making the bacteria make that protein for us. Okay? And so now insulin's relatively cheap, right? And it's more accessible to most patients. And so this is just what I was talking about, right? Because all these processes on every cell in the planet, including bacterial cells, is extremely similar, we can do this in bacteria. So in order to understand how you go from here's DNA that we PCR'd out to here's the insulin protein, we're going to learn a little bit about that process. The process of going from the instructions, the gene, to the actual product, the insulin. Okay? And it's a little more complicated than just going from DNA to protein. There's an intermediate molecule called RNA. Okay? It's very similar to DNA, but its job is to transfer information and then be used to make protein. Right? So the central dogma of molecular biology or molecular genetics is that our DNA molecule codes for an RNA molecule, which then codes for the actual protein. Right? So this would be the gene. This is the intermediate information. This is the insulin. Okay, the product. So DNA to RNA to protein. We can never go DNA straight to protein. Every single cell on this planet uses this process of going from DNA to RNA to protein, right? Whether it's a bacterial cell or a human cell, or an amoeba, paramecium, algae, anything, every single cell, okay? Okay. So genes, which we've been talking about this whole time, are just little parts of a chromosome, right? The DNA sequence in a part of a chromosome, A, G, C's, and T's, right? The sequence, the order of all of those in a specific region makes up a gene, right? So we know chromosomes are all this double-stranded DNA. You know, so if we unwrap it, it's super, super long. A small part of that chromosome would be one gene. So the gene for insulin. And then next door to it would be another gene, the gene for some pigment for your eye color. Next door to that might be the gene for DNA polymerase. Next door to that might be the gene for your earlobe shape. That's expressed during development. Okay? Each chromosome has thousands of genes. Right? So a gene would just be a small unit, small segment of that long DNA molecule. Okay? And remember, there's only those 4 different DNA nucleotides, the A, G, C's, and T's. It's the number of those and the order that they're in that determines what the gene makes. Right? So every gene has a different sequence, a different order of these nucleotides. So we can think of excuse me, the nucleotides as analogous or very similar to the same kind of thing as letters in the alphabet. Right? The same 4 letters, if they're in a different order, can mean something completely different. Right? They're the same stupid 4 letters, but the order is what determines whether you're writing the word fart or the word raft. And as it turns out, those are 2 different things. If you're on a deserted island, you don't try to find a fart or build a fart, okay? Or you'll be deserted forever. Okay? So it's just the same as with genes, right? There's only the A, G, C's, and T's, but GGG, TTT, CCCC, AGCT codes for something different than a bunch of T's, A's, and C's, right? It's that order that determines it, which is why it's so incredibly important for DNA replication to copy each strand exactly correctly. That whole stupid idiot proof, A's and T's go together, G's and C's go together. Because that order matters. If you mess up the order, that's a mutation, right? And then you might not make the right RNA intermediate transfer molecule, which means you won't make the right protein. You might make a form of insulin, but it doesn't work right because you have a missing nucleotide or one too many or the wrong one somewhere in there, right, to make that protein. It's happening. Okay. So then here's just a picture. They're trying to illustrate, I know the concept of the difference between chromosome and gene and DNA can be very confusing. It's even confusing to our majors. And so they're showing us this little part of chromosome 1, right? It's all wrapped up in its little sausagey shape, just so we can see it. But really, it's this big, long piece of double-stranded DNA, right? Which are just, right, the little rungs in the inside are A's, T's, G's, and C's, that whole sequence. And they're just showing us that a small part of the chromosome, maybe 1,000 base pairs, 1,000 of those nucleotides all linked together, is a gene. And each gene is the instructions for building one protein, usually one protein. Okay? So the example in the book, the story is about antithrombin, which is a gene that helps with blood clotting or keeping your blood from clotting. Okay? My example is always insulin because I think that's something people can understand what that end product protein does. Okay? And gene expression is just something that's expressed as something that's turned on. Right? If every single one of our cells has all 46 chromosomes, your skin cells have the gene for insulin. But skin cells don't make insulin because that gene is turned off. The DNA's there. It's sitting there. We just don't put the switch on. Okay? But all of our cells have all of our genes because they have all of our 46 chromosomes. It's just whether we turn them on or turn them off. Okay? And that's all gene expression means, whether genes are turned on or genes are turned off. And so then our little picture, right? Again, gene expression, if you turn that gene on, right, it will make that, you will create that RNA intermediate molecule, which we'll talk about how that works a little bit. And then from there, we can synthesize a protein to do the work. So their example is the antithrombin protein would be the same for insulin. Insulin gene would only be turned on in your pancreas in a specific cell type and no place else in your body. It's the only place it's supposed to be turned on. Okay. Whereas DNA polymerase, the enzyme that does DNA replication, would be turned on in all of our cells. Right? We might need to replicate every single cell type in our body. So that gene would be turned on in all of our cells. Okay? Most genes are only turned on as a few specific cell types. Right? If you're making liver enzymes, you need to be a liver cell. Those genes are only turned on in your liver, not in your skin, not in your stomach, not in your brain cells. Yeah. Yes. Woo! So the question is, if there's a gene turned on in a cell where it's not supposed to be on, can that cause cancer? Yes. That's what does cause cancer usually. Usually a gene that's supposed to be off gets turned on. And it's usually a gene that helps control cell division, making more cells. Right? Sometimes it's a gene that's supposed to be on that gets turned off. Right? A gene that's on that's in charge of not letting cells divide. If you turn that off, and that lets them divide. Right? Or some other gene that is supposed to be repairing mistakes. You turn that off. Now you don't repair mutations. Now you get mutations in genes that turn on and cause cancer, cause the cell cycle to go, go, go. Right? So appropriately controlling gene expression, or where genes are turned on and off, is absolutely necessary for survival. Right? If things are turned on and off wrong in development, the embryo dies. Right? Everything needs to come on and off exactly right for that one single cell to become a ball of 8, to become that stupid blastula that sticks on the side of the uterus, that then becomes that tiny little embryo that then forms all the organs, right? If genes turn off when they're not supposed to, huge developmental problem, embryo dies. Or turns on when they're not supposed to, developmental problem, embryo dies, right? So this process of controlling when genes are on and off is absolutely essential for survival. Yeah. The question is, can scientists manipulate mutations in genes? The only kinds of mutations that they can manipulate would be like the gene therapy stuff we were talking about, and very few things work for gene therapy, right? If we're making, we can manipulate mutations in genes, like if we do PCR for the insulin gene, and we found a way to make insulin more active, we could make a change in that in vitro in the test tube, grow up a whole bunch of it, and make like insulin on steroids to be better for diabetics. That we could definitely do.
Thank you very much for coming today. I will see you on Thursday.